Good afternoon, everyone. It's a pleasure to welcome back our Board on Life Science and also all of those of you that are watching this as a webinar. We have a very interesting presentation, and it's going to be on the frontiers in the microbiome-gut-brain science, which is a very exciting topic that a number of folks have worked very diligently on. So the goal of the discussion is to identify fundamental science and technological gaps and needs in neuroscience. It's going to focus on the interplay between the brain and other physiological systems, specifically the microbiome-gut-brain axis. During the discussion, speakers will be asked to describe trends in their fields, particularly as it relates to the microbiome-gut-brain axis, trends in wearable technologies to measure and manipulate brain activity, and effects from the environmental conditions on the microbiome. And so with that, I'm going to turn it over to Kavita Berger, who will introduce our moderator, and then we will proceed.
Thank you very much. We are very pleased to have you join us today for this discussion. By way of introduction for our moderator for today, Diane DeIuliis is a distinguished research fellow at the National Defense University. She has spent quite a lot of her professional career working in and out of the life sciences, national security issues, policy and preparedness related issues, and she has a degree in neuroscience and started her career off in the National Institutes of Health working on neurodegenerative disorders. And so with that, I would like to turn your attention over to Diane.
Great, thank you, Kavita. And I will just say a quick apology for being 1 or 2 minutes late. Still, occasionally there's a challenge with getting on these Zoom calls. So hopefully you guys can hear me and see me okay. So once again, thank you so much for the intro, and thank you for the invite to moderate this discussion. I think it's going to be really interesting. There's going to be a variety of information and data that I think is very rich indeed. The committee on that's been working on biotechnology, I've been participating in the ad hoc committee to that larger group, and we've had some really interesting dialogues about the microbiome and about everything that's been said in introducing this session. So I don't want to take away from the speakers, and I will confirm with you, Kavita, that we are slightly departing, I believe, from the written agenda as to the order. And so I want to confirm that Anu is going first, correct?
Correct.
Okay, got it. So I will briefly introduce Hanumantha Kathasamy, who is a neuroscientist and neurotoxicologist in the Department of Physiology and Pharmacology at the University of Georgia. He has recently been recruited to the University of Georgia at Athens as the John H. Isaacson Chair and Georgia Research Alliance Eminent Scholar in Parkinson's Research. As you know, Kavita, during her intro, discussed that I had some background in this space when I was at NINDS, and I'm keenly interested in what Anu has to present. He has been working on Parkinson's and other neurological disorders, and has worked in the interdepartmental toxicology graduate program, has served as the president of a neurotoxicology specialty section, and has a lot to relate to us today. So without any further ado, Anu, I'm going to turn it over to you for your presentation. Thank you.
Thanks, Diane. We could have my slides. All right. Can everybody see the slides okay? Is it not a full mode?
Yes, they look great. Thank you.
Right. Okay, thanks for the opportunity to present some of the recent trends that's going on in the field, particularly as related to neurodegeneration in terms of the brain, gut-brain microbiome axis. As Diane mentioned, I've been recruited to UGA recently to set up a center. If you could move to the next slide, on a Center for Neurological Disease Research. A little background about the center. That was in honor of Dr. late Senator Isaacson. He was passed away about a few months ago with Parkinson's. And the Georgia, University of Georgia and the state has invested some new initiative to set up a center on neurodegenerative diseases. as a past part of the initiative, there was initially 2 faculty, and I was recruited to hire 4 more and completed the hiring the last 1 year. So we have totally 6 faculty working on kind of 4 research thrusts. One of the main one you see in the left side of the translation discovery, we put the microbiome metabolic engineering as the main focus, how that new technology could be addressed to potentially develop some new strategies, understand the gut microbiome interaction, the gut-brain axis. And then also we have a lot of neurotoxicologists in the center. We have recruited about 3 neurotoxicologists already we have within the university, 3 or 4 more. So about a group of 6 neurotoxicologists will be working on how environmental risk factors would potentially play a role through the gut-brain axis. The next slide, please. So just to orient everybody to the disease, it's as you know, the Parkinson's is an incurable neurological disease. It can affect anybody at any stage. You know, some of the figures listed in this pictures. The late dissection, the genocidal reaction is the latest victim of the disease. Since the discovery of the clinical description of the Parkinson's disease in 1817, in the last 200 years, we learn a lot more about the disease in terms of the pathology, neurochemical basis, the neurocircuitry involving extrapyramidal motor control systems. So basically what happens in the disease, about among billions of neurons, only a few thousand neurons in substantia nigra degenerate. That's all dopamine-producing neurons. The loss of the dopamine causes impairment in the extrapyramidal motor circuitry, leading to motor symptoms that manifest as a tremor, rigidity, akinesia, and postural instability. That's often referred as a trap. And also there is a non-motor symptom that precedes the motor symptom. I'll come back to in a moment. In terms of the pathology, in addition to loss of neurons, there is intracellular inclusions that contain some misfolded protein like alpha-synuclein. That's been a lot of interest in this area of understanding why the protein misfolding, as well as some using this as a biomarker. In terms of risk factors, the aging is an important risk factor, as like any other disease. There are other risk factors like traumatic brain injuries, environmental chemical exposures like solvents, and heavy metals like manganese and lead, and pesticides, PCBs. That's been linked to Parkinson's range. In terms of odds ratio, there is a 3 to 4-fold increase in the risk if you have any of these exposures. In terms of gene defects, there is about 20 different genes been identified since early 1980s. And then you have these gene defects like synuclein, LRRK2, GBA, PINK1. Those gene defects contribute so 10% of the disease, till 90% we don't know what's causing the disease. The hypothesis is environmental factors a dominant risk factor for the disease. All these stressors, environmental stressors and genetic stressors, drive the disease through oxidative stress or protein degradation pathway, leading to inflammation and degeneration. Some unresolved challenges in the field are understanding the mechanism, particularly gene-environment interaction, and potentially we don't have any diagnostic biomarkers, trying to develop diagnostic markers, and also therapeutic strategies in terms of disease-modifying therapies. Next slide, please. So in terms of the time course of the disease, you can see the symptoms, before midpoint where you see the symptom starts, there is a long prodromal or a preclinical period that takes about almost more than a decade. So that the silent period also is challenging. It provides an opportunity. If we can make intervention at the early stages, then we can slow down the process. And the late stage, when you have the disease diagnosis, then there is effective treatment currently to control the symptoms, levodopa. That drug is effective for about 6 to 7 years, then causes severe dyskinesia. That's also, we want to develop some strategies to reduce the dyskinesia. I'm going to give you some examples of how that the L-dopa and the dyskinesia can be reduced using some microbiome based therapeutical approach. And then the early stages, I'll give you some examples how the metabolites could contribute, potentially to inducing some of the disease processes. So some of the symptoms, early ones are you'll see that the gut dysfunction, constipation, one of the earliest symptoms that's related to also some sleep disorders, RBD sleep disorders, or potential risk factor. The RBD, once people diagnose RBD, the recipe conversion is for 90%. So then you lead to hypoxia, then some other autonomic dysfunctions, then finally leading to motor dysfunction, as well as at the end you will have dementia and psychosis. Next slide, please. So in terms of environmental factor, there is internal as external factors. Internal factors include aging, genetics, lifestyle, diet, and health. And the external environmental factors includes pollutions, solvents, metal exposure, and pesticide, as I mentioned earlier. These convergence of these two factors, so these two factors can contribute to the disease processes. So I'm going to give you some examples of those, each of these areas. Next slide. So what we found is when you analyze the blood samples and the urine samples, we found one of the metabolites, it is untargeted metabolites, we call trimethylamine oxide. Then we took the metabolites, and then it is pretty high in our Parkinson's patients, used the cell-based, animal-based models. We saw some increase in protein synuclein aggregation. Thus, the synuclein protein aggregation is a part of the disease process. We found some in the cell-based model that is triggering the inflammatory response. Next slide, please. Interest of time, we can talk a little bit more, but I'm going to give you the general overview. So what we come up with the hypothesis that this project is funded by DoD to show that when you have a choline and beta-diene, many people take high choline, high beta-diene food, if that not metabolized properly, then you have a higher accumulation of TMAA, the trimethylamine, and then it's further metabolized in the liver as a trimethylamine oxide that further catabolizes into formaldehyde and trimethylamine, dimethylamines. These metabolites can interact with alpha-synuclein and then form a protein aggregation in the brain. That's an area that we're looking at making some interesting data that coming out soon. Next slide, please. And then in terms of environmental exposure, that's I showed you example of the internal metabolites. These external toxicants like manganese. Manganese is a heavy metal. Exposure to manganese been linked to Parkinsonism, either through drinking water or welding, occupational exposures, or working in the steel manufacturing industries, where you see there is, when you add, treat the animals with the manganese, you see some gut inflammation, as well as in the inflammatory marker like INOS and TNF and IL-1 beta levels were increased. Next slide, please. So that leads to also some metabolomic changes in terms of the some large short chain fatty acid, as well as some of the bacterial. We saw some changes in Firmicutes and Proteobacterium during chronic treatment of this metal exposure, showing that some changes as a single metal concentration can affect the gut metabolites as well as the microbiome, cause dysbiosis. Next slide, please. Next slide. So this is a very interesting strategy. What I would like to do is show you that one of the main problem with Parkinson's disease, if you have take this the levodopa, the drug is quite effective for about 5 to 7 years. But later part, it causes some dyskinesia. There is a video, I don't know whether we can click the video on that person that's sitting in the chair. So that person, when you have that L-dopa, around 6 to 7 years, they cause severe dyskinesia. The problem there is you have to give the tablets like 4 to 5 times a day to keep it as a stable to be effective. So where it causes a pulsated delivery, so you have every time you give a tablet, that plasma level goes up, then comes down. So over time, the therapeutic window narrows. At the time, off time increases, that also causes dyskinesia. So the system, we want to keep any neurotransmitter level as stable. Unfortunately, we cannot be able to do that with the current treatment. So we hypothesized that if we can deliver L-DOPA stably through the gut bacteria, then we should be able to reduce the dyskinesia. In order to do that, we were able to screen and find a couple of enzymes that should be able to, they can convert the tyrosine from the food to the dopamine. What happens in the brain, you have tyrosine is converted to dopamine, L-DOPA, and for the dopamine by tyrosine hydroxylase. We were able to identify two enzymes that are capable of converting tyrosine and then clone that into the E. coli Nissel bacterium. This is the probiotic bacterium used to quite safe for over 100 years. We use that E. coli Nissel as a chassis and then put rhamnose as inducibles. We can titrate them tunable so that dose response manner, we can control the drug delivery and put it in the animal as a proof of concept. And then we did some pharmacokinetics and pharmacodynamic studies. Next slide, please. So here is we use a model called MitoPark. If there is certain whether you can click that MitoPark slide. You will see some videos, but maybe the video is not working. If you could click that, please. Yes, so that's working. So this model, the MitoPark model, is the transgenic mouse model. This model, 8 to 10 weeks, looked normal. What we did in this model is we knocked down the mitochondria only in the dopaminergic neurons. So after 16 weeks, immediately they develop some locomotor deficits. And then around 24 weeks, they become highly Parkinsonian. That also correlates with the neurochemical changes and loss of dopaminergic neurons for 90% lost around 24 to 30 weeks. So we use this model. And then if you click the bottom one, we fed that bacteria to this mouse model. Then we were able to see that they're able to have a more locomotor activity. So then we did some pharmacokinetic studies to show whether we can measure the plasma level L-DOPA. What you see in this next figure over there is the therapeutic windows. When the bacteria, our bacterial administration stays at therapeutic level with a single dose, whereas the chemical L-DOPA, the darker line, you will see that increase immediately shoots up, then comes back within 6 hours. When you give this bacterial delivery twice a day over a 10-day period, you can do stable level of L-DOPA. You can also increase in dopamine level. Then when you do some behavioral assays, what I showed you before, this unbiased behavioral assays, open field, water maze, and post-swim and tail suspension. So we found this animals do better in the locomotor activity, even some memory functions is improved, and the depression test, they perform better, showing that decreased depression. Because we are administering catecholamines, you would expect to see some increase in their reducing anxiety, increase in performance level, overall performance. Next slide, please. So that work was funded by NINDS. The previous slide in the translational program, we are trying to advance this to currently to primate studies and also for additional the dyskinesia model, L-DOPA dyskinesia model. We are testing that bacteria to see whether we can take that approach to the clinics. Since we saw increase in cognitive function, then we also tested the same bacteria in the Alzheimer's model. Also, the L-DOPA, the next step, when you have the dopamine pathways, L-DOPA converted to norepinephrine. So the norepinephrine, noradrenergic neurons in the Alzheimer's degenerate much more early than cholinergic neurons. So we reason that the increase in norepinephrine and the dopamine level would probably improve memory function in the Alzheimer's mouse model and rat model. So we tested both in the APP knock-in as well as the rat model of Alzheimer's. We saw improved in the cognitive function. And then also there is reducing the amyloid load. We think that the increase in the catecholamines, they can stimulate stimulate the immune cells in the microglia and astrocytes to increase the phagocytic activity and scavenge some of the amyloid load. So we think that this approach not only provides the as a neurotransmitter, replenishing neurotransmitters, it also can probably modify the disease over time. So this work is funded by Alzheimer's Drug Discovery Program. We are trying to test that in the multiple models and two or three different labs to kind of unbiased approach as a blinded study design to see whether this we can generate enough preclinical data to advance this work. Next slide, please. So then we went on to test whether mouse model, whether translate to higher model. So we did some pharmacokinetic studies in dog model. Beagles, about 3-year-old beagles, we tested about 1 month, 2 different dose level, dose escalation studies. We saw about 700 to 800 nanogram level of L-DOPA levels, showing that the drug increases L-DOPA level to a therapeutic level in human. And also that you can wash off, then the level comes back, as well the dopamine, the neurotransmitter level, as well as the bacterial counts. We can measure them in the fecal content. So we're using this dog model. One of the observation when we review the literature, beagle dogs, about 70 to 80% of the beagle dogs develop cognitive deficits. It's called canine dementia in dogs. So that the dementia model that includes also amyloid pathology. When you do the studies have shown, there is a cognitive dysfunction in this dogs that correlates with amyloid deposits. So we are going to test this after running all these studies in the mouse model. One of the aim is to translate that to the higher model, the dog model, to see whether we can improve the cognition, also reduce the amyloid load in the dog models. Next slide, please. So we did some metagenomic studies in the dog fecal material. We saw we didn't see any significant differences in the gut bacterium, showing that this strategy doesn't affect drastically the gut bacterium. There are some minor changes. We are trying to understand what are the changes. Early indication shows this may even improve the gut microbe population to a little more healthier bacterial community there. So we thought, we also found there is some the L-DOPA metabolizing enzymes, also one of the bacterium we were able to normalize. We are trying to further analyze the data. Next slide, please. So this is overall strategy for the translational work, showing that L-DOPA genes can be cloned into the bacterium, then that what we call as a live bacterial therapeutics that can be formulated, then ultimately administer to the patients to improve the neurotransmitter over time. So kind of showing that the gut bacterium can be used as a kind of drug manufacturing factory that could produce its own drug over time, so that we don't have to administer, that allows a very stable level of neurochemical changes in the brain. Next slide, please. Next slide. Maybe it's not. So think they have one more slide, I think. So anyway, there is, I think, some challenges and opportunities in this area that we can discuss at the end. This technology has a lot of excitement. At the same time, there are some bottlenecks we need to overcome. With that, I will stop here and take questions, or maybe you can wait at the end.
Okay, thank you so much, Anu. This is fascinating stuff. As you know, I have a very keen interest in this. It's just fascinating to me, the direction that this research is going. Kavita tells me we should get through all of our presentations first so that we can summarize the discussion for at the end. So I'm going to quickly introduce Olivia O'Leary, who is a senior lecturer and principal investigator in the Department of Anatomy and Neuroscience, University College Cork, where she leads a research program investigating the neurobiology of stress-related psychiatric disorders like depression and looking for novel targets for therapeutic development. So she's going to talk us more about gut microbial regulation and how neurons in the hippocampus are affected by this and the implications it has for cognitive and stress-related brain disorders. So Olivia, while we're getting you started with your slides, I want to remind everyone, if you are on the live stream, that you can put questions and comments into the Slido. For those of you in the room, please hold your questions because we know you have some for Anu. And Olivia, I'm going to turn it over to you. Thanks.
Great. Thanks very much, Diane, for your introduction, and also for Kavita for the invitation. It's really wonderful to be involved in a panel discussion like this. I'm looking forward to the discussion later on. So, oh great, yes, I can see my slides. Thank you very much. So as Diane has alluded to, my research focus, if you go on to the next slide, please. My overarching, I suppose, research interests over my career has been really focused on unraveling the neurobiology underlying stress resilience, depression, and antidepressant action in an effort to identify novel therapeutic targets for the treatment of stress-related psychiatric disorders such as depression and different types of anxiety disorders. Next slide, please. So much of my research focus has been really heavily focused on the hippocampus area of the brain. The reason for this is that the hippocampus is a very important structure in regulating the response to psychological stress, also behavioral responses to antidepressant drugs. In turn, the hippocampus itself is also very sensitive to negative impacts of chronic stress or traumatic stress. So, for example, neuroimaging studies in humans have consistently reported reductions in the size, so volumetric reductions in the hippocampus in individuals with post-traumatic stress disorder or depression. And then finally, the other key, the other reason why I'm interested in this brain structure is because it's obviously the key learning and memory center of the brain. And we know that under stress, learning and memory processes don't work as efficiently as they might do. Next slide, please. So what really is fascinating about this brain region, however, is that it is one of just two areas of the adult brain that contains neural stem cells, which we know can produce new neurons. It's debated how late in life this can occur, but we know that it can occur in humans, and it does occur in many mammalian species. So lots of these new neurons are born, but the majority of them don't survive. So this, the rate of production and the survival of these new neurons and how they mature and then eventually become integrated into the neural circuitry of the hippocampus is influenced by various intrinsic factors, but also extrinsic factors, which I've summarized there in the diagram. Related to my interests, we know that chronic stress or chronic exposure to stress hormones, corticosterone, cortisol, decrease hippocampal neurogenesis. And we also know from animal models that chronic, and also actually in humans from post-mortem studies, that antidepressant treatments, including a variety of antidepressant drugs, but also other therapeutic strategies, like, for example, electroconvulsive shock, increase hippocampal neurogenesis. And we know from animal models that adult hippocampal neurogenesis is required for at least some of the anxiolytic effects of chronic antidepressant treatment to be manifested. Hippocampal neurogenesis also plays a role in certain types of learning and memory processes, in particular spatial learning and memory, but also a process that's called pattern separation. So pattern separation is the ability to distinguish between very similar experiences or contexts or environments. So, for example, if you go to your office every day and you park your car in the same car park as do all of your other colleagues, but each day you park your car in a different space. Well, most of the time, most days, you remember where you parked your car. Impairments in this cognitive ability for pattern separation, however, is an early symptom of age-related mild cognitive impairment. And more recently, there is evidence emerging that it may also be impaired in stress-related disorders. So taken together, this process of hippocampal neurogenesis plays an important role in stress resilience and also in cognitive function. And therefore, any, I suppose, interventions that can manipulate this process may have therapeutic potential for stress-related disorders and cognitive brain disorders. So next slide, please. So one such intervention would be manipulating the gut microbiome. So many years ago, I hypothesized that that hippocampal neurogenesis may be shaped by the microbiome. So to test this hypothesis, we investigated different stages of hippocampal neurogenesis in male germ-free mice. So these are mice that lack a microbiome, and they're reared in a microbe-free environment. And indeed, we found that there were alterations in various aspects of hippocampal neurogenesis in these mice. And since then, other groups have also reported alterations in hippocampal neurogenesis in germ-free mice on different genetic backgrounds. It has also been reported that antibiotic-induced depletion or disruption of the gut microbiota in mice can decrease hippocampal neurogenesis. And there are individual reports reporting that some bacterial strains or combinations of certain bacterial strains can also influence hippocampal neurogenesis, either positively or negatively. Next slide, please. So the key question for us really is, well, how can the microbes in the gut actually influence this process in the brain? And one of the key candidates is the vagus nerve. So the vagus nerve transmits neural information bidirectionally between the gut and the brain. And it has been shown that some bacterial strains can induce antidepressant or anxiolytic-like effects in animal models via the vagus nerve. So if the vagus nerve is severed, then they can no longer induce these beneficial effects. So this is true of some bacterial strains, but not all. Intriguingly, stimulation of the left afferent vagal nerve at the level of the neck, the cervical level, is an approved treatment strategy for treatment-resistant depression. So this really reinforces that vagal nerve signaling to the brain can regulate mood and emotion. Next slide, please. So we had shown that previously that severing the vagus nerve through a surgical technique called subdiaphragmatic vagotomy, by doing this in animals that have an intact microbiome, that this actually reduces adult hippocampal neurogenesis and also reduced the protein brain-derived neurotrophic factor, which is an important protein that supports the growth and the survival of neurons. However, what's currently unknown and what we're kind of working towards is whether the vagus nerve itself mediates gut microbial regulation of hippocampal neurogenesis and neurogenesis specific behaviors. So, for example, pattern separation, And in addition, whether this occurs under different contexts, so a different environmental context, for example, under stress conditions or during aging. Next slide, please. So this diagram is just to illustrate, however, that there are many other routes other than the vagus nerve that through which gut microbes could potentially influence hippocampal neurogenesis. So these include endocrine mechanisms, for example, modulation of the stress axis, the HPA axis, components of the immune system, for example, monocytes, T cells, and the microglia, which are the immune cells resident in the brain. And also very complexly is the potential of metabolites, so the products that bacteria produce as they are metabolizing our dietary components. So in addition to investigating the role of the gut of the vagus nerve, we're also investigating the role of key microbial metabolites in the regulation of adult hippocampal neurogenesis. Next slide, please. So there are lots of microbial metabolites to choose from. So the microbiota really is a powerhouse for producing loads of different very useful substances, ranging from vitamin B to neurotransmitters. And I'm sure Rima will talk a lot more about this. But some of the metabolites that we're particularly interested in are short-chain fatty acids. So short-chain fatty acids are produced by gut microbes as they metabolize dietary fiber. And these short-chain fatty acids include acetate, propionate, and butyrate. And very interestingly, some of these short-chain fatty acids, in particular butyrate, has the capacity to inhibit enzymes called histone deacetylases. And we know that histone deacetylases are very important in regulating the expression of genes. So we're very interested, and we're currently investigating whether these short-chain fatty acids may inhibit specific histone deacetylases within neural stem cells, and in turn then alter hippocampal neurogenesis. However, these microbial metabolites, as I say, they're produced locally in the gut. There are receptors in the gut for these microbial metabolites. So we're also interested in whether, the possibility that these metabolites are interacting with receptors and vagal nerve afferents, and then in turn altering vagal nerve transmission to the brain, which might then in turn alter hippocampal neurogenesis. So it's very likely there's more than just one avenue for gut microbial interaction with hippocampal neurogenesis. Next slide, please. So why is this research important? So unlike the hippocampus and the rest of the brain, where drug accessibility is limited by the blood-brain barrier, the gut microbiome is highly accessible to direct interventions. So for example, prebiotics, probiotics, dietary interventions, even fecal microbial transfer. So I suppose the overall goal, or the overall hope, is that by directly targeting or manipulating the gut microbiome, this in turn could perhaps indirectly alter neural activity, which may be difficult to achieve through normal drug development processes. However, to achieve this, we really need to understand the mechanisms by which specific gut microbes and different types of gut microbiomes can alter brain and behavior. And then in turn, this may reveal non-invasive precision therapeutics for brain disorders. Next slide, please. So of course, this comes with huge challenges, and I'm sure this is going to be a discussion point later on. And these challenges are captured very nicely, I think, in this perspective piece by my colleague John Crine and Sarkis Mazmanian. So there are cross-species differences in brain and behavior. So obviously the findings in animal models may not necessarily translate to humans. In turn, it is very challenging to try and recapitulate human genetic diversity and the varied environmental influences in human life into animal models. And while there are human studies that clearly demonstrate there is alterations in gut microbiome in various disease states, and that there is potential for different products or bacteria to alter neural activity, these studies tend to be small and correlative. So the human studies are really lacking behind in terms of mechanisms compared to the animal studies. So what's required really moving forward, I suppose, in the translational world is is large-scale human longitudinal studies. These would inform if microbiota alterations precede a clinical diagnosis or vice versa. Such studies would need to include the impact of the relationships between gut microbiota, host gene and host environment interactions by including control like shared gene controls or shared environment controls such as household controls or sibling controls. And of course, include the big data. So include multi-omics analysis of both the host and the microbiome. And just recently in Nature, a very nice paper came out using ingestible devices to sample the gut luminal contents for metabolomics and various other omics approaches. So coupling these multi-omics approaches, perhaps with neuroimaging and tracking host behavior in detail, so host behavior and host physiology and host diet, I think perhaps through the use of wearable technologies is really going to push the field forward. So with that, I'll leave it to the next speaker. Thank you very much for your attention.
Okay, thank you so much, Olivia. That was fantastic. And thanks for covering so much so quickly. Again, everyone, please sort of remember your questions and what Olivia had there. Some of the big picture topics on that end slide are some of the things we can include in terms of speaking about challenges and how to push the fields forward during our discussion. I'm going to go ahead now and introduce Eran Blacher, who is an assistant professor in the Alexander Silberman Institute of Life Sciences at the Hebrew University of Jerusalem in Israel. And he did his first postdoc position in the microbiome-brain interface at the Weizmann Institute in Israel, where he was one of the first to study the role of the microbiome-gut-brain axis in the context of neurodegenerative diseases. Personally, I find it fascinating to even begin thinking about how the gut and diet can affect neurodegenerative diseases in general. And there's so many different pieces to this. He's going to talk to us about a bunch of different techniques, including the integration of imaging, multi-omics, advanced computational analyses, and clinical observations. So I will turn it over to Eran.
Hello, everyone, and thank you very much for the kind invitation. I'm very excited to be here and to share with you some of our latest findings. So my name is Eran Blacher, and I study the gut-brain communication in aging neurological diseases. I will briefly today present you some projects that we are invested in on gut-brain communication. Next slide, please. Throughout my career, I expertise in neurobiology, microbiome research, and aging research. My primary focus now is to understand how the aging gut influences neurological disorders and how this can be translated into therapeutics. Next slide, please. And next few clicks. Thank you. Okay, so in my lab, we integrate knowledge from different signaling hubs in the human body, the nervous system, the gut and its microbiome, and the immune system. And the special thing about those systems is their ability to sense signals from the environment, process them, and relay that information to other systems and organs in a way that may shape our physiology. Next slide, please. So I feel personally that we experience a special period in biomedical research, and I hope I'm not exaggerating by calling it a paradigm shift, but we neurobiologists are definitely shifting now from the brain-focused dogma to a much broader holistic approach to neurological disorders, looking at the crosstalk of between many organs and systems. Next slide. And next few clicks, please. Thank you. So to frame my discussion today, I will very shortly touch three exemplary projects. First is about the importance of integrating aging and circadian rhythms in brain science. The second is about the gut microbiome, the metabolites it can modulate and produce, and how they affect neurodegenerative diseases. And the third project is about the gut barrier in stroke, barrier integrity, and post-stroke inflammation. And I will try to use those examples to stress out where things in the field are heading, the needs, the gaps, and state of the art. Next slide, please. Thank you. A few clicks. Thank you. So when we talk about the immune system and the gut and how it affects the brain and vice versa, most of the people in the field are trying to understand what makes us sick, what are the pathogens, the cell types, the genes, and the pathways involved. And I argue that it is at least as much important as to ask when we get sick, because most of our physiological processes are rhythmic and oscillate across the time of the day. So if we work, for example, on mice that are nocturnal and test behavioral and physiological features only during the day, we can bias our observations. And this may be one of the reasons why so many therapeutic targets for neurological disorders that were identified first in mice fail later on in clinical trials. Next slide, please. Thank you. So accordingly, we ask what happens to our innate immune system when we age. And we isolated to answer this question macrophages, which are dominant immune cells of the innate arm, from young and aged mice every 4 hours across 24 hours. And one of the most remarkable things that we have discovered is that almost 700 genes showed rhythmic transcription in macrophages across the day in young mice, but only 50 genes in aged macrophages. So circadian transcription was dramatically disrupted in aged macrophages, with the majority of transcripts losing rhythmicity, as you can see on the second, in the middle heat map here. With aging being the biggest risk factor for so many neurological disorders, we must consider circadian rhythms when we test specific genes and how the pathways they regulate affect physiology. Next slide, please. And few clicks, please. Thank you. So in the next project that I describe now, we discovered we basically tested the connection between gut microbes and amyotrophic lateral sclerosis, ALS. And we discovered in mice, in a mouse model for ALS, that specific bacteria were able to produce nicotinamide, a metabolite that can ameliorate ALS in this model. Next, we transitioned to conduct a small observational human study, and we recruited ALS patients in Israel. And I want to use this example to describe how we designed this clinical trial, because we used here family members as controls, individuals that share the same household with the ALS patients. So as you can see here, we found that even though we use these very stringent control conditions, by integrating into the research control individuals that share the same environment, same kitchens, for example, or facilities, we indeed see significant different clustering of their microbiome composition and function. And they also presented significantly lower levels of nicotinamide, as we saw in mice, in their serum and cerebral spinal fluid. Next slide, please. And next few clicks. So just to summarize this project, we saw that in mice that were basically littermate controls, wild types, and ALS model mice, the SOD1 transgenic mouse model, they developed different microbiome compositions early on, even before clinical symptoms occur. Next click. Few clicks. Thank you, and one more. So by treating these mice with Akkermansia muciniphilia, a microbiome species that is significantly reduced during disease course, we were able to replenish the amount of nicotinamide secreted to the systemic circulation of these mice. It can reach the spinal cord where it can modulate the activity of motor neurons and help them better survive the stress and delay disease symptoms such as skeletal muscle degeneration. And I listed here on the left side a few of the methods that we used to accomplish this project that was very long, just to show you how many different modalities we need to integrate in order to get a meaningful insight and establish the causal link between specific microbial composition and a given physiological phenotype. In this case, it's ALS progression in mice. Next slide, please. Few clicks. Thank you. So in microbiome research today, we see more human-focused studies than 5 years ago. We see larger cohorts and longitudinal sampling, as was stated before. And we use more sophisticated techniques to profile the microbiome, such as shotgun metagenomic sequencing. So we get the entire DNA content of a given sample, rather than just the 16S microbial ribosomal subunit that tells us which bacteria are in the samples, but not what are their functional potential. Next few clicks. Thank you. We integrate more controls to make our studies more sound and stringent. For example, when we study in mice, we use different housing conditions. We look at different vivaria to see if our findings repeat in different environments. And we overall transition from a very descriptive way of basically cataloging bacteria and describing them into a more mechanistic research that aims to establish causality between the microbiome, its composition, the functions that it mediates, and phenotypes such as many different features of our physiology. And luckily, we have many ways and many means to manipulate the microbiome in order to validate our results. So we can wipe the microbiome out, or at least part of it, by using antibiotics. We can use germ-free mice that are devoid of any microorganisms, and we can colonize them with specific microbial species that we can isolate. And now we expanded our ways to grow anaerobically in vitro more and more bacterial species of the microbiome, something that was not so easy a few years ago. We co-house different strains of mice, which is a more physiological way to transfer microbiome from one strain to another. We can, of course, perform fecal microbial transplantation, FMT, and use metabolites and pre and probiotics as therapeutic approaches. Next slide, please. Thank you. And the third project that I will describe is about stroke. Stroke, again, is is one of the biggest causes for death and disability in elderly populations. And we know that myeloid cells are metabolically impaired with aging. Macrophages, monocytes, and other myeloid cells that originate from the bone marrow, they are less efficient in using glucose and shunting it into the TCA cycle and producing ATP and energy. And this leads to a disruption in immune homeostasis and resolution of inflammation. And again, we study stroke, and we discovered that in aging, the injured brain sends a stress signal mediated by the sympathetic nervous system to the gut that hits the gut and leads to a significant, prolonged and sustained gut barrier breach. And then bacterial molecules and bacteria themselves activate lamina propria macrophages in the gut. Next few clicks. Thank you. These pathogen-associated molecular patterns and the actual live bacteria can invade the bloodstream and further facilitate brain damage. And our aim is to metabolically reprogram those innate immune cells to rejuvenate the gut mucosa and to fight this post-stroke inflammation that affects basically the whole body. Next slide, please. So the biggest, why this is so important, because the biggest problem with stroke is the amount of time that takes to reach the hospital and revascularize the injured brain by surgical or pharmacological means. However, we have very limited control over these factors. On the contrary, the post-stroke inflammation unfolds for weeks to months after a stroke, which gives us a very wide window for therapeutic intervention. And therefore, it is very important to study this post-stroke inflammatory process in the context of aging. Next slide. So we integrate aged mice as a model system to study brain diseases. We use the distal middle cerebral artery occlusion in these mice as a model to induce stroke. In this model, you can appreciate that the infarct size, the injured area of the brain, is larger in aged mice compared to young mice. And we follow these mice throughout stroke progression, before and after. Next slide, please. We discovered when we looked at the guts of these mice that the aged mice experience a very profound gut barrier breach. They have leaky gut that lasts 3 to 4, even 5 days after stroke. That gives a very long period of time to the immune system to be activated and to cause more and more damage to the periphery and the brain. Next click. And as you can see here, in aged mice, we can detect lots of pro-inflammatory markers in their systemic circulation, in this case, in their serum. Next slide. Thank you. So before I finish, I would like to talk a little bit about technology. When we study the gut, and I guess this is true to lots of other organs as well, but as a scientist who is focused on the gut, I can tell you that location matters. So there are many different niches in the gut, vascular niches, neuronal niches, the mucosa, the lining, the epithelial niche, lymphoid follicles and organs, the mucus layer, and of course the microbiome. And it is important to unravel patterns of cell-cell communication in which localization is important to understand more about the function of the cells, not only the expression levels of given genes and metabolites, for example. Next few clicks. So in our lab, we integrate spatial biology. We have multiplexed imaging system that allows us to image 100 plus markers simultaneously in a given tissue, in the gut, in the brain, and we correlate those features together with other disease-related data. I think this is definitely one of the ways this field is heading. This approach is very common now in oncology, and I guess in the next few years we will start seeing more and more gut-brain focused researchers integrating these spatial biology systems more and more. Next slide. And just to summarize, there are many risk factors that are associated with aging and therefore also with neurological disorders, atherosclerosis, hypertension, dysbiosis, which is a pathological change in the microbiome composition and function, inflammation or age-related inflammatory response that sometimes is subclinical, but it lasts for many years. High body mass, hyperglycemia, hyperlipidemia, and sleep disorders. And we try to integrate at least some of these comorbidities in our mouse models, and we try to consider them when we model neurological diseases. Next slide. We can skip this slide for the sake of time. Thank you. So to summarize, our future goals are to, in the lab, are to identify novel players and interactions in gut-brain intersection, to unravel the aging gut's contribution to disease. And I think many of the causes for neurological diseases are lying within the gut, and to open a breakthrough approach to treating specifically post-stroke inflammation and maybe in the future other neurological diseases as well. With this, I would like to thank you again for your listening and participation, and we'll be happy to take your question later. Thank you very much.
All right, thank you so much. So we've got 2 speakers left and we have 25 minutes. So let's hopefully get through these other 2 speakers, because I do want to make sure there's some time for discussion. I know that some of you may be able to go up a little past 2 o'clock, maybe. Unfortunately, I cannot, but Kavita will take over for me in that case. So without further ado, let me introduce Todd Coleman, who's been just so much fun to work with. He is the chair of the National Academy Standing Committee on Biotechnology Capabilities and National Security Needs, where we have discussed many of these microbiome issues related to the nervous system. And I won't go too far into it. It's been fun to watch good arguments between Kavita and Todd on Michigan go blue he has in his ongoing statements. And Todd has, and he's got a sign behind him. That's perfect. Kavita can be annoyed by that later. Todd, I'm going to turn it straight over to you to get started. Thank you.
Thank you. Thank you. And I think the most important part is the Q&A. So I'll move fast. So I thought what I wanted to do by start is to tell you a little bit about myself and my trajectory so you can understand my perspective. So I started out with a PhD in electrical engineering with absolutely no interest in anything biological. My PhD advisor strongly encouraged me to do a postdoc in something wildly different. So if you advance, this led to me doing a postdoc in neuroscience with Emory Brown. It was an experience that I call feeling comfortable, feeling uncomfortable, because I didn't know anything about neuroscience at the time, but I was able to learn physiology through the language of probability and statistics. And then as I began my career, I got involved in some technology development. Next slide. And so early on in my career, next slide, I got involved in some things where not just doing the standard applied probability stuff, which is an exciting time now because of big data, but also involved in technology development to do things like measure physiology from the human body with unobtrusive sensors. And from the neuroscience perspective, the focus was very much on the brain. So for instance, a patch that can track your sleep with just a patch on the forehead. Next slide. Then something happened where as a picture of me and my dad at a family reunion, and I lost him to pancreatic cancer. And that process, it turns out he had lost his mom to stomach cancer years before. So I got to thinking about trying to do something involving the digestive system, even though I knew nothing about it whatsoever. So the story of my talk is feeling comfortable, feeling uncomfortable. I challenged myself to do that again. Next slide. And so when I first began to think about the digestive system, I was a little worried at first because I'm an electrical engineer / brain scientist / neuroscientist. I think about the digestive system. I think about the acids in the stomach. I'm not a chemist. I think about the motility of pushing stuff through the digestive system. I don't study mechanics, but then lo and behold, I learned that there are pacemaker cells throughout the lining of the digestive system, and they are and these pacemakers are connected via the nervous system in our gut that has 100 million neurons, and that nervous system is actually connected to our brain electrically. So there's actually an electrical and a neurophysiology story here. So I began to be more intrigued. So next slide. So the high level vision that we got interested in is building on what the EEG has done for the brain, what the EKG has done for the heart, and can we build a non-invasive device that can track the electrical patterns of the digestive system in humans. Next slide. And so we have been able to do that in a series of publications over the last decade. And so we've been able to build, unobtrusive technology like a patch that you place over the abdomen that can track the electrical patterns of the digestive system. We've developed novel signal processing algorithms that we can track the spatial propagation of the slow wave of the digestive system, and in particular, not only in human controls, but also patients with disease. And most importantly, we've been able to showcase in patient studies that, features of the spatial patterns that we cover electrically correlate with the symptom severity. And one finding in particular was the first finding that was done non-invasively in humans for patients with upper GI disorders like delayed gastric emptying, which occurs quite often in Parkinson's patients, for instance, preceding their motor symptoms. And this was the first such measure that actually correlates with the severity of symptoms. So next slide. So if you click advance again, everything I just told you about for this last decade was primarily focused from the perspective of gastroenterology and trying to help gastroenterologists develop better ways to understand the cause of patient symptoms and treat them. But in the back of my mind, I'd been thinking about the brain because of this electrical and neurophysiology story. So one thing that really got me excited, next slide, is a finding. So first, let me just tell you a little bit about the electrical coupling between the brain and the gut. So imagine when you eat food or something like this, the distension of the stomach or certain nutrients being processed, that actually elicits a sensory mechanism, just like photons hitting the eye, or likewise with the ear. And the sensory mechanism first goes through brainstem areas, such as the nucleus tractus solitarius. And the first cortical area that it hits is the insula, and in particular the posterior insula, which is a sensory part of the insular cortex. and then that passes information upstream to the anterior insula. And along with going through many parts of the brain, this actually sends downstream information to brainstem, nucleus tractus solitarius, and then dorsal motor nucleus of the vagus. And then it sends a downstream motor response to modulate aspects of the digestive system activity. So there's this nice electrophysiologic coupling between the brain and the gut. Next slide. So one of the things that got really exciting to me was that someone showcased that you could capture this coupling in humans. And so this was a group out of France in 2017 that did simultaneous monitoring of the electrical patterns of the digestive system using the same approach that I did, the electrogastrogram. And that signal is a very slow oscillating signal, about 0.05 hertz associated with the stomach. And they simultaneously recorded electrical activity of the brain and electromagnetic activity, I will say. So they use a technique called MEG, which is capturing the magnetic fields of the brain. And what they were able to showcase is that in certain parts of the brain, the oscillations that you see, their amplitude is actually modulated by the phase of the slow rhythm of the stomach. So the takeaway lesson here is that this is an inhuman, non-invasive measure of the coupling between these two organs. Absolutely fascinating when I found this. Next slide. It's the good news. Bad news is that MEG is not as ubiquitous as other types of measures of brain functioning. For example, in 38 states in America, there's no clinical MEG location. So click advance. Not surprising what someone would think about doing. We thought, well, hey, could we pick up this same type of gut-brain coupling if we replaced the magnetic field with the electric field? Meaning, can we use EEG? Next slide. And so the original study from 2017 showcased certain brain areas where you see more red. Those are the areas where a specific band of the EEG, in particular the alpha band, around 10 hertz, was coupled, and its amplitude was coupled by the phase of the slow signal from the stomach. So that's that picture plot. And then we showcased last year, advance, that you can pick up similar types of information if you use EEG. In one area in particular is this right insular area as I'm pointing to. Next slide. So I showed you this, electrophysiologic coupling between the brain and the gut, but how does this relate to aspects of the microbiome, which is the topic of our discussion, advance? So there's a nice review paper from Science by Elaine Shao and her research group, and they talk about many aspects of the gut-brain axis within the context of inflammation, but just within the context of neuronal aspects, interestingly, if you click advance, some of the areas showcased in her diagram include the nucleus tractus solitarius and dorsal motor nucleus in the vagus that I talked about before. And if you click advance, some key high-level points, and other people talk about this before me, is the short-chain fatty acids and the free fatty acids produced by fermentation that occurs in the intestine. This affects permeability of the blood-brain barrier and neuroinflammatory responses. But interestingly, also via enteroendocrine cells, this directly engages the enteric nervous system and up to the central nervous system via some of these neurophysiologic electrical pathways. In addition, the gut microbiome, their metabolic products affect things like GABA, motilin, and ghrelin activities, which we know are tied to hunger signaling and whatnot. And these then modulate central nervous system driven effects on meal responses, as well as emotional responses. So very exciting aspects that are at play here. So moving forward to the next slide. This is a high-level conceptual point, and actually other speakers already talked about this, that as we start to see about the ways that these two systems are interconnected, me as an electrical engineer, this is not surprising, but you can imagine if these two systems share variables, rather than trying to go through complicated ways of perhaps directly electrically stimulating the brain by cutting a hole in the skull or trying to get drugs to pass through the blood-brain barrier, could we imagine directly modulating these pathways via the gut, via things such as manipulating the microbiome or appropriate drugs, or even possibly electrically stimulating peripheral organs and using this neurophysiologic pathway to get to the brain? So that is the high-level story, and what I would like to do is have ample opportunities for question and answer. So with that, I'd like to pass the microphone back, and I'll talk about that in Q&A if necessary.
All right, great, Todd. Thank you so much. Okay, so let's move on quickly to Rima. and who is a professor in the departments of psychiatry and behavioral science and medicine at Duke. And she has been working in the space of neurodegenerative disease for some time now, looking at metabolomics. And she actually co-founded the Metabolomics Society and has been more than 4 years as its founding president. So I'm going to pass it over to her quickly so that we can try and maximize the time at the end for questions. Thank you, Rima.
And thank you, Diane, and many thanks to the organizers. We are going to make this very brief and very, just highlights of lessons learned. If I can have the slides, please. Yeah, lessons learned from large initiatives funded by NIH and mainly by the National Institute on Aging. The next slide, please. We're just going to move fast and highlight concepts. These are the large consortia funded by NIH, including the Gut Microbiome Initiative for Alzheimer's disease, metabolomics for a whole area of neuropsychiatric disorders. So we are going to highlight lessons learned by scaling up our understanding of metabolism, right, and using metabolomics as an enabler for precision medicine approaches for the study of CNS disorders. The next slide, please. How we view the world is as follows. We know that the genome does influence our health. We know that. We know it is not the only influencer, right, for inflicting disease and some of these disorders. The exposome where we live, the air we breathe, the water we drink, where our address matters, the social interactions, sleep patterns, socioeconomic status, and the gut microbiome. All of these imprint and modify metabolism in profound ways. And what I'm showing on the right side is the end result of all of this is an impact on our metabolic state. And therefore, in the next slide, as a metabolomics community of researchers that are interested in a precision medicine approach and in capturing the net influences of what we described in the previous slide, we are measuring thousands of chemicals in the blood, in the brain, in different tissues, right? And we are expanding the horizon from a magnification of 1x to 10x to 100x and beyond to measure a very rich array of chemicals that exist within our system. Some of it is, in the next slide, please, is certainly influenced by our genome, right? We are creating atlases with our partners in Germany, where we are studying the influence of the genome on the metabolome. But more recently, it has become clear that the gut microbiome, the bacteria that coexist with us, you have heard a lot about some chemicals produced by the bacteria. I can tell you that humans and the bacteria complement each other's, complement metabolism in some incredible ways. What you see on the upper right-hand side is a concept that we can complete metabolism of bacteria. Some of the things that the bacteria ingest from what we take in the food produces chemicals that we can further metabolize, and the bacteria can metabolize things we produce. So it is a true partnership between a human and bacteria that produces an incredible number of chemicals. And the tip of the iceberg was neurotransmitters we looked at from the GABA to the serotonin and on to sulfur-containing compounds, branched-chain amino acids, bile acids, and so on. It's only the tip of the iceberg in an amazing number of chemicals that are either produced or influenced by the gut bacteria that coexist with us. So the metabolome gives you a read on the influences of the genome and the gut microbiome. The complexity gets in when we realize that the food that we ingest modifies the gut microbiome, modifies the metabolome. So these are interacting factors that cannot be studied in isolation. Truly, if one wants to study the gut-brain axis, what we eat matters, the exposome and what we are exposed to in the next slide. In the next slide, drugs we take, supplements we take, environmental chemical exposures are all things that do modulate the gut microbiome and the metabolome, all of which can influence the gut-brain axis. And therefore, we are building big databases by working as research communities, connecting databases on foodomics, databases we are being created on drugs, drug metabolites, supplements, all of it, the influence of it on the gut microbiome and the metabolome is being investigated. Adding to all of this is environmental chemical exposures from pesticides and onwards. So we are talking about interacting factors that truly affect our health, and it needs to be taken into consideration as a collective, co-regulated systems or co-influence systems that can influence the brain. In the next slide, taking all of this, we are building databases. We don't have time to talk about enabling research communities, overcoming barriers to enable communities at large, learn more about these gut bacterially produced chemicals. We have released a MIME database with Dr. David Wishart, a resource for the community to learn something about these bacterially produced chemicals. In the next slide, the key is, is to understand this coexistence and the co-regulation between bacteria and human, a field that truly was pioneered by Nathan Price, who is with us in the audience, Ina Steele, and many others. So we are applying a whole array of tools to try to better understand this bacterial human co-metabolism and model it. In the next slide, we want to understand, then taking a big snapshot view, what's in our brain in terms of chemicals, right? And what about this gut-brain axis? Or is it the liver gut-brain axis? Or is it the kidney liver gut-brain axis? I tell you now, after studying CNS diseases for 20 years biochemically, there are no strict, clear boundaries around these disorders. Parkinson's, depression, Alzheimer's, right? Anxiety, there's a lot of things that are common. It's a whole spectrum of metabolic alterations. And I can tell you there's a whole array of chemicals produced by bacteria that we can clearly now see in the brain. So we are mapping the brain metabolome, the influences of the exposome, the diet, the gut microbiome. And there's a whole array of chemicals without having bacteria in the brain that we could detect. We are able to see a whole array of chemicals produced by bacteria. In the next slides, again, we have very short time. A large funded initiative by NIA is the Alzheimer's Gut Microbiome Project that became expanded to the exposome. Just few words quickly on what this initiative is. In the next slides, we bring 40 researchers to work together, from engineering to biochemistry to metagenomics, gut bacteria and clinical, put it all together to try to understand changes in the gut microbiome related to cognitive and neuropsychiatric symptoms in Alzheimer's patients. Depression, anxiety, cognition seems to be all related metabolically, biochemically, and with a link to the gut microbiome. We work as research communities, large 10 Alzheimer's centers around the country. We have big engines that were put together, NAC, NICRAT, SAGE, to be able to put the data public. We have kits that go home to patients wherever they reside across the United States so that we can get fecal samples and very shortly, I hope, blood samples that can flow in a low-cost way that can expand the horizons and bring samples so we can analyze changes in the gut microbiome, changes in the metabolome, link it to cognition, link it to changes in mood. We also study the influences of diet and lifestyle. A big project focuses on bringing studies like the MIND Mediterranean diet, ketogenic diet, the pointer, the lifestyle, a subdivision of the finger. The lifestyle and the diet is the biggest influencer probably on the gut microbiome, on the metabolome. We are connecting all of this with cognition. In Project 3, we model all of these interactions by manipulating the gut microbiome in animal models to try to think about mechanisms in a deeper way and to try to come up with novel therapeutic approaches. Moving forward, just a couple of examples of what we see chemically as a chemical community. We can skip all of this. Let us go to chemical communication and what we are learning. We can skip this, please. Let's move on. Give you 2-3 examples quickly on, let's go back, please, one. Here is an example of cholesterol metabolism, which we know is implicated in Alzheimer's disease in a big way. What was not studied is the cholesterol clearance to produce primary and secondary bile acids by bacteria in the gut, and the realization from a whole array of studies in thousands of patients where we discovered that the secondary bacterially produced bile acids from cholesterol clearance are connected with cognitive changes, brain imaging changes. And the interesting thing is that they are found all in the brain, and a partnership also with the Institute of Systems Biology, part of Nathan's Price team, then illustrated that these secondary bile acids probably are not produced in the brain. They are transported from the blood to the brain, and they correlate significantly with these cognitive changes. not only in the next slide, not only do we see a correlation between these bacterially produced bile acids as a relationship to cognition, but a very strong link to anxiety. And the second speaker highlighted elegantly the importance of the biochemistry of the bacteria in mechanisms of anxiety and depression. Here we are looking at a whole array of chemicals produced by gut bacteria, secondary bile acids, that very strongly correlate with anxiety. And the tools that one needs to connect the gut and the brain is imaging. We can go back now in the next slide, be able to use imaging data, including the study of executive control networks, and be able to illustrate that a change in the bile acids in the blood reflects major changes in the brain, right? There must be this communication at multi-levels, but this biochemical communication is one of them. And the examples can go on. Next slide. We can go into not only the bile acids is the tip of the iceberg. We are discovering thousands of bile acids with Dr. Peter Dorrestein, illustrating the opportunity and the gaps in the field. What we are studying today as 40 bile acids might end up being thousands of bile acids produced jointly by bacterial and human co-metabolism. And ending up, we can look at the indoles, another example just to illustrate that this is not only a bile acid story. The next slides, please. Indole metabolism that ends up being in the gut, serotonin. Tryptophan metabolism produces serotonin, produces kynurenine. But what you see in the orange is a whole area of metabolism of indoles in the gut that we sometimes forget to study. And as it turns out, the metabolism of tryptophan in the gut produces a whole area of indoles. We can skip a couple of slides just to show you that indoxyl sulfate, a product of indole metabolism, depending on what bacteria you have in the gut, you can produce protective indoles or cytotoxic indoles. The indoxyl sulfate is produced by bacteria and kidney enzymes in the kidney that sulfates bacterial products. A high level of the indoxyl sulfate strongly correlates with anxiety, correlates with fMRI changes in patients who suffer depression and anxiety. And we can move on and manipulate now the indoles in model systems. Last slide, maybe we can skip all of this. In this slide, no, one slide back, we can now go to model systems with the researchers at Stanford and at Caltech, and be able now to engineer bacterial strains that are overproducers of the indoxyl sulfate, underproducers genetic manipulations, and we can clearly see behavioral changes in these animals. I think the chemistry matters. The area of chemicals produced matters. Doing community research is a big enablement, big power, making our science public, right, which is the NIA mandate. Not only are we working as communities together, we are making our data public in real time, has changed the paradigm in how we view science, how we view the brain, and the brain disorders are no more diseases of the brain. They are systemic diseases, truly, that affect the brain, affect the whole array of other organs, and connecting now these other organs through this biochemistry might be a phenomenal approach that brings an integrated view, a precision medicine approach to the study of CNS disorders. I'm going to stop here. Thank you.
Oh, thank you so much, Rima. That was fantastic to have you wrap all that up at the end. And I think that there's been a recurring theme throughout all of these discussions, right? Which is the complexity of tying all these different fields together, right? So not only just looking at the systemics of gut-brain interactions through the nervous system, and all of that implies, including genomics, epigenomics, all of that affecting it. But then also all of the diagnostic approaches, the clinical approaches, how do you measure these things in real life patients? What's the meaningful science there? So given that this is so complex, I think the big question, which I'm going to start in the room first, and I think Kavita is going to take over for me when I have to leave in a minute or two, is to kick off a discussion of how do we bring this field, how do we bring these studies forward, and how do we make progress in this arena based on everything you've just heard from these excellent speakers. So we can go to the room first and hear from the life science group there. And then I know there's some questions from folks online too that hopefully you can get to after the folks in the room can ask their questions. Over to you, Kavita.
Thank you so much. So while our board members are thinking about questions, I will say that we are going to maybe go long about 5 to 10 minutes, but not eat in too much of your time because I know some of you have hard stops. For our board members, please feel free to raise your hand or drop a question in the chat. We're happy to ask. But I do want to start with where Diane had left because the complexity here is quite interesting. And the degree to which many of you use techniques all the way from whole organism to, the highly molecular to be able to understand what is going on and the pathway of what is going on is quite interesting. And so one of the questions that we had as we were sort of chatting back and forth was, what are in your mind the biggest sort of scientific challenges or hurdles that need to be overcome to be able to continue to further this research, to continue to look at it from a more systems level and to really understand the direct or indirect consequences? So any of our panelists can start.
Oh, maybe in my mind, I had initially 10 minutes, I rushed through my talks, but I am very excited about the engineering approach, right? So you have, if there is a dysbiosis or the metabolites deficits, then if you have a autonomous sensing system coupled to engineered bacterium, so you sense and deliver, right? So that type of approach would be very exciting. So I think that's kind of the emerging the next 5 to 10 years. If there is a dysbiosis in the metabolites, a dysbiosis in the gut bacteria associated with the metabolites, there are circuits that can sense and they deliver required metabolite. That would be very exciting and see how that changes phenotype.
Thank you.
I think
Rima, go ahead.
from my perspective, removing barriers between fields, from engineering to physics to clinical to biochemical, truly has taken us in a profound way forward, thanks to NIA and the vision of NIA, right? And then thinking about the system, the whole system, these organs crosstalk, removing boundaries among diseases. What we are finding is many of the diseases have things in common, especially the neuropsychiatric disorders, removing boundaries, rethinking the textbook of medicines, the textbooks we study is going to be important to bring changes.
Excellent, thank you. Todd or Olivia or Ron, do you have any thoughts you want to share?
This is Todd here. Two things come to my mind. First of all is, as I mentioned a little bit about the electrophysiology and the neurophysiology, there are people that do that, and then there are people who do a lot of other types of studies. I have not seen a lot of figuring out the right way to blend those together to get 2 + 2 = 5. So I think that's an opportunity. We need to think it through. Something else that I think would be interesting is trying to develop some characterization of, depending upon the context. So I think what a lot of us talked about are interesting ways that we could potentially manipulate one organ to then modulate another. There are many ways we could go about doing that. And, is there some fundamental way that we can understand how much specificity can we get at achieving our target using one approach versus another? I don't really see that yet. I see people working on sort of their preferred ways of trying to do that. But do we have enough knowledge of the physiology and the mechanisms that we can really understand in a context-specific fashion when one approach may be superior to another? So those are my 2 cents. Yeah.
Thank you. Iran or Olivia?
Yes, this is Iran here. I completely agree with Rima. I would just like to add that big collaborations and cross-continental collaborations, for example, looking at different lifestyles and different nutritions that people have in different geographical areas may help us to increase the knowledge and to strengthen the links that we may find between disease and lifestyle. So these big consortia are a very good opportunity to achieve this information.
Thank you. Dominique.
Yes, hi. Broad question for all of you. Fascinating research. Thank you for the presentation. Can you say a word about the bioethics around microbiome research, particularly as is related to brain research?
Rima, do you want to start with that?
Yeah, and I think, I'm thinking about the metabolome. We are facing the products of the bacteria that imprints part of the big metabolome, and making metabolome data public, there are issues. It does identify us. Not only is our genetic makeup an identifier, we are seeing clearly we are unique metabolically, and some of the data we are putting public could identify individuals. Each one has a unique identity. So I think the question is right on, and we need to think about it as we make our data public, right? The issues, of course, of identifying individuals and protecting individuals is key.
In the bioengineering side, there is a lot of bioethics issues, the biosafety issues. Suddenly, if you engineer a bacterium, the genetic modification, how you control the biosafety, and as well as the, the biocontainment, the horizontal transfer. Those are some bottlenecks that need to be looked into. I think there are some tools that various fields are developing that could be incorporated into the gut microbiome arena.
Thank you. So we have two questions that have been shared via Slido, and I want to ask them both and hear from you what your thoughts are as our final questions of this session. So one of them is about the role of organoids in advancing this research, and a question about whether any of the methods that you are using or any of your colleagues are using, whether they include organoids, in vitro organoids. And then the second question is actually the question that draws on some of the broader discussions about diversity and inclusion, in that, do you see any gender differences in any of the interactions between the microbiome, brain, gut, and its effects? And one of the sort of questions that we end up sometimes thinking about are, what are are there differences in between genders, between populations, between various different communities, in their kind of health and overall health and well-being, based on the environment, based on what they're eating, based on various different other issues. And so want to throw both of those questions out there to you and see what your thoughts are. And maybe while you're thinking about those, I'll let Scott ask his question.
Thank you so much. Thank you for those wonderful presentations. I had a quick question for Dr. Dauk. That was a very interesting presentation about this individual identifiability from metabolomics. I mean, isn't it already the case that sufficient data for individual identity is available on genomes, for example, on NCBI, even if they're anonymized? In other words, you can still probably figure out there's a unique sequence. And so does the metabolomics issue differ from that situation somehow?
Not really, but it was a surprise for us that metabolically we are also unique, as much as we differ among, and differ as we change our lifestyle and diet, we change our metabolomes, yet there remains a unique identity. I look like myself more than I look like anyone else. So the issues are similar to the genetics. May I just comment on the gender difference is remarkable. Analyzing the bile acid metabolism from a human and bacterial co-metabolism, men and women come from different planets. The dysregulation that we saw that was so profound in Alzheimer's disease afflicted more men. In this case, more men specifically, the perturbations than women, whereas the story with other aspects of neuropsychiatric symptoms differed. So I think there are certainly major differences between men and women metabolically, and part of it is imprinted by the gut microbiome.
Thank you. Okay, Val, you get the last question.
Thanks. Yeah, let me just confirm. So when you note that the microbiome is unique, all of the genome, I wonder, and this is just an ignorant question on my part, is it changing such that it's not static? So when we say that it's unique, the genome is give or take, from a plain language, non-scientific perspective, I think perceived as somewhat static. Is the same true for the microbiome? Because to Scott's comment, what I heard was that the issues around privacy and ethics and confidentiality and agency and things like that were not unique, because they more or less mirror the issues that arose with regard to genomics. But it crosses my mind that there is a potentially distinct feature if the microbiome changes over time.
Todd, did you want to answer that question, or did you have other things that you wanted to talk
I mean, I don't.
I wasn't going to answer that question, but can people hear me?
Yes. I'm not
I
sure.
was going to go back to the question about diversity and gender specifically. I was just going to say that there's a big unmet need within the context of the gut-brain axis and reproductive health. So if you take a look at across the phases of the menstrual cycle and whatnot, and a lot of so we just step back for a moment. If you just take a look at GI problems, for instance, something I know more about, it's a lot more represented in females versus males. Now, if we focus a little bit on some of the things that occur throughout the phases of the menstrual cycle and how that overlaps with the gut-brain axis, I haven't seen nearly as much science on that as you would think you should see. So I think that's a huge emerging opportunity. Yeah, that was my comment.
And Rima?
A quick comment, Valerie. The genome is stable, the metabolome is unstable, and so is the gut microbiome. So indeed, it changes as we change our lifestyle, as we exercise, as we change diet, and so on. But the interesting thing, there is still a unique biochemical identity for each. And the only time we lose some of this identity is when we get afflicted with a disease. We start to see, right, some changes, but there is a unique identity in spite of the dynamic nature of the, at least from the metabolome perspective. We have, there are more experts than me on the gut microbiome, but the metabolome is dynamic and yet is a unique identifier.
Thank you so much. So we are hitting about 15 minutes over, and we have two hands up. So what I'll do is I'll have both Lucilla and Olivia sort of make their comments, and then we will close the session and thank everybody for joining. So go ahead, Lucilla.
Yeah, just quickly regarding re-identifiability, there is also the problem of linkage of data sets across. So that is another area that I think needs some caution about making everything public.
Thank you. And Olivia?
Yeah, just a comment regarding the sex, biological sex differences and gender diversity. But one of the issues in our field with the animal models is that a lot of the behavioral tests have been developed in males. And a lot of the preclinical field of gut microbiome regulation of brain function and behavior is biasing towards the males, in that it seems to have, the gut microbiome seems to strongly more influence neurodevelopmental outcomes in males rather than females. But by the same token, a lot of the behavioral tests that we use to assess these things in the males animal models, they're not suitable for females, or the tests themselves, behavioral tests themselves, have not really been validated in the females. So I'm not entirely convinced in the field that sometimes this report of male bias is actually really accurate until we develop these good animal models that are suitable for both males and the females. I think only then we're going to get some answers.
Thank you so much. And thank you all for joining us today. This was fascinating. Obviously, there are lots of scientific and social questions, as well as sort of experimental questions that remain for us sort of in this field of science, and as we broaden to looking at other kind of multi-system systems, I guess. So I want to thank all of our speakers today for joining us, and we look forward to engaging with you in the future.